Assalamualaikum and hi, we are from group 32. We present about advances in non-destructive DNA extraction from teeth for human identification. This is our team members. My name is Nurhanis Akila binti Muhammad Nawi and my matric number is 20246899324. So, what is tooth? Tooth is a hard and calcified structure in mouth that help to bite, chew and break the food. It is also important for DNA identification. Let's look at the tooth anatomy. Tooth consists of crown and root. Tooth also made of 4 tissues which are enamel, cementum, dentin and pulp. First, enamel is the outer layer of crown that consists of 96% of mineral. This makes tooth the hardest tissue in the human body that acts as a protective barrier. But it does not have DNA. Cementum, which are the outer layer of root, which consists of 70% of mineral. That will give cementum a dense and low porosity structure that limits the penetration of microorganisms that can cause degradation. We can also obtain DNA from cementum. Third one is the dentin part. It is hard because it is also made from 70% of mineral. The dentin lies between enamel and cementum and acts as a protector from damaging the pulp. It is also contain nerves that transmit stimuli and are responsible for absorbing pressure from chewing. Pulp does not have mineral contents, but it is rich with blood vessel, nerve and connective tissues. It is also function as a protector from infection, sensor temperature changes and pressure, produce new dentin and helps keep the tooth moisture. Why is teeth important in forensic? Teeth is useful in forensic due to its protective structure. It have outer layer which are enamel and dentin that protect DNA from environmental damage, which are decomposition, degradation, microbe attack and high heat. It is also a non-destructive source of nuclear DNA even after a decade of death. It is also used for identification in forensic cases. Tooth is the last surviving biological material in a mass fatality cases. It can survive in an extreme condition that can destroy other evidence like hair and bones. Examples are fires, tsunami, and plane crashes. We can use a non-destructive extraction to retrieve DNA without damaging the tooth by using modern technique. We are also able to preserve its archaeological and morphological value. Example forensic case that use teeth for DNA identification is case missing tourists in Himalayas. A furry went missing in a dense forest in Himachal Pradesh, India. Months later, sheep head from fragmented skeletal remains. Teeth collected during autopsy were analyzed using a powder-free DNA extraction method and compared with blood sample from the missing man's wife and son. The outcome is the identity confirmed with more than 99.99% probability of paternity.
Hello, I'm Joanne and I will be presenting about the DNA preservation in teeth and why cementum is the best source. Every tooth tells a story, but not every part of the tooth tells it equally well. In forensic biology, knowing where DNA hides inside the tooth and why it survived can be the difference between identifying the victim and never knowing who they were. When a person dies, the body begins to break down, and inside the teeth, not all tissue respond to the same way. Different parts of the tooth preserve DNA very differently from each other. In fresh or recently deceased individuals, pulp and dentin still contain good quality DNA because the tissue haven't had time to decay yet. But in age, degraded or old remains, the kind forensic scientists most often deal with, only cementum can rely upon to preserve DNA. This is exactly why modern forensic DNA research has shifted its focus entirely towards cementum. So what makes cementum so special? The answer lies in its structure. Cementum contains cells called cementocytes, and these cells are locked inside tiny space surrounded by a dense mineral matrix. Think of it like a natural vault. This vault protects the DNA from 3 main tricks: the enzyme that would break it down, the bacteria that would consume it, and the chemical decay from the surrounding environment. Unlike dentin, cementum has much lower porosity. This means fluid and microbes simply cannot penetrate it as easily, keeping the DNA safe inside. And here is something fascinating: as the person gets older, the cementum layer actually gets thicker. So more cementum means more cementocyte, which means more DNA present. Look at the side by side. Cementum, high DNA preservation, high STR success, the best option especially in aged teeth. Dentin, moderate to low preservation. Its tubular structure allows fluid to seep in over time, making result unpredictable and variable. Pulp. Despite being cell-rich in fresh teeth, it performs poorly in aged remain. It has no hard protective matrix, so it is one of the first tissue to break down after death. The table speaks for itself. When it's come to old or decayed remains, cementum is the only tissue you can truly rely on. But even cementum is not invisible. 2 categories of factors can affect how well DNA is preserved. First, environmental factor. Acidic soil accelerates DNA breakdown through hydrolysis. High moisture encourages microbial growth that degrades DNA. High temperature speed up chemical damage. And the longer the burial time, the greater the cumulative damage to the DNA. Second, tooth-related factor. Permanent adult teeth or proven baby teeth because they have thicker, more developed cementum. Dental disease like tooth decay can compromise cementum integrity and reduce its protective capacity. But interestingly, older individuals actually have an advantage here because their cementum is thicker, storing more DNA. This is why forensic scientists must carefully evaluate each tooth before attempting any extraction. So why does all of this matter? In real forensic cases, disaster victim, war casualties, missing person, the remains are often old and severely degraded. By that point, pulp has decayed, dentin yield nothing useful. Cementum becomes the last and only reliable source of nuclear DNA available for identification. And that choice, knowing which tissue to target, is what determines whether a family finally gets answer about their missing loved one or whether the case goes forever unsolved. In forensic biology, the right tissue makes all the things difference.
Hi, my name is Nurhanis Atika binti Muhammad Nawi. I'm going to explain to you about the extraction methods used in this study. There are 3 of them. The first is destructive method. The tooth was broken, crushed and ground into powder. The DNA then was extracted from pulp, dentin or cementum. The advantages of this method is it provided high DNA recovery yield and useful for very old or damaged remains. The 4 disadvantages of it are the tooth is completely destroyed, the morphology cannot be studied later, ethical concern for historical or cultural remains, and higher contamination risk. The second method is partially destructive method, which are pulp drilling and cementum scraping. Pulp drilling happens when a small hole is drilled into the tooth to collect pulp tissue. Pulp drilling causes less damage than total destruction and moderate DNA recovery but leaves visible hole. Pulp may already be degraded and have variable success rate. While on the other hand, cementum scraping is when the outer layer on the root surface is scraped gently to collect the DNA-rich material. It produces moderate to high DNA yield, cause only minor surface damage and faster procedure. But the operator's skill affects the result and aerosol contamination may occur. The last method is non-destructive method. The tooth is not being cut or broken. The DNA was released from the cementum surface using a chemical incubation. Tooth was cleaned, tooth was soaked or incubated in extraction solution, and the DNA diffused out without damaging the structure. This technique preserves tooth morphology, low contamination risk, suitable for museum and archaeological specimens, and allow future analysis, but require longer extraction time and other disadvantages. Are the comparison table for each of the techniques, and they have their own advantages and disadvantages.
Hi, I'm Nur Shafika Asmi, and now I will briefly discuss the comparison between teeth and Petrus bone as DNA sources for human identification. The journal explains that Petrus bone, which is part of temporal bone in the skull, is considered the best for highest DNA recovery because its dense structure protects DNA very well. However, extracting DNA from Petrus bone is highly destructive as it damages the structure of the skull bone. In contrast, teeth, especially dental cementum, provide a minimally destructive alternative because teeth are highly durable and can still produce reliable DNA profile while preserving the physical structure of the specimen. Therefore, teeth are preferred when ethical preservation and conservation are important, while Petrus bone is mainly used when maximum DNA recovery is necessary from severely degraded remains. This method has several important forensic applications. The first is human identification. To identify unknown individuals, particularly in burned, decomposed, or skeletonized remains, where other soft tissue are unavailable or degraded due to environmental damage. Because teeth are highly durable and can preserve DNA for long periods. The second is disaster victim identification. In cases involving plane crashes, fire, tsunamis, and war-related incidents, because teeth may survive this extreme condition even when other tissue are destroyed. This allows DNA recovery for dental comparison and legal documentation. The method is also valuable in missing person investigations where preserving the remains is important for further analysis such as isotope analysis and morphological studies. Moreover, this article strongly emphasizes ethical consideration. Unlike traditional DNA extraction methods that often involve drilling or crushing teeth, with permanently damaged human remains. In contrast, this advanced non-destructive method help preserve valuable evidence and respect cultural and religious concerns. It also highlights the importance of scientific integrity and contamination prevention, where strict contamination and laboratory control procedures are necessary to prevent DNA contamination and false identification to ensure reliable results. In conclusion, this article demonstrates the importance of non-destructive DNA extraction from teeth in modern forensic science. This method balances between effective DNA recovery with ethical preservation of human remains. Overall, this method improves forensic investigation while promoting more ethical forensic practice for the future.